Abbiamo altri 3 o 4, 5 minuti? Sei già partito tu col streaming?
Worldwide streaming.
Io lo seguo con lo streaming.
Sei già partito? Non so, aspettiamo altri 5 minuti. Aspettiamo 13 minuti ancora. Stiamo ancora 2 minuti. Non è che girano la funzione native di Linux, cioè è Linux, penso sia una soltanto c'è come OS. Se poi adesso stanno dando supporto a applicazioni che girano dei container, cioè non girano un sistema operativo.
Forse questo li posso arrivare alla partecipazione.
No, secondo me loro non vogliono proprio che tu non vogliono avere il sistema.
Ma si riesce ad avere una scelta marginale?
Sì, ma è stralimitato. C'è una scelta, non ci può fare il censo. No, se voi installi, loro lo chiamano Linux System, però sono dei container.
Sì, ma hanno tagliato cose, hanno customizzato bene e giustamente lo sappiamo. Cosa che nessuno
abbiamo scoperto alcune famiglie come no, questo non funziona perché questo qua mi sta fare questa è la share sua. Questa di sua è limitata.
Se è contenuta,
Il bash, dove qualcosa fa.
sì, ma è contenuta nel senso anche se ha però
no, tu devi stranità.
però non c'è. Io ho provato con questa scusa se io mi sto appropriando no, dove ti fanno il browser e il browser dà l'accesso parziale al sistema
però per esempio in questo c'hai il sistema operativo ottimizzato per tutto eccetera, e se poi vuoi installare MRTX qua funziona anche per dire perché si sta dentro il container è limitato. No, per esempio funziona mentre ad esempio sul sistema bash, cioè sotto Windows 10 che invece penso sia una macchina virtuale, vabbè possiamo discutere sulla differenza tra macchina virtuale e container, lì non girano per esempio
le macchine dipende anche il livello di virtualizzazione
che qua X Windows System di Chrome OS, mentre su Windows devi installarsi Xload come si chiama X3. Ok, allora facciamo è partito? Ah, ok. Okay, so welcome to the second Open Science Seminar for 2019. Today's speaker is Matteo Caffini. He got his PhD in physics in 2010 at Politecnico di Milano. Since then, he always worked as a researcher and data scientist in the field of medical imaging, specifically focusing on neuroimaging. He's currently a postdoctoral researcher, part of the Animal Cognition Lab in Rovereto. And today he will be talking about structural data for multimodal imaging in an open research environment, the functional near-infrared spectroscopy case. Floor is yours.
Thank you very much. So, a little premise. This is going to be probably my last seminar as an academic. As a few of you may know, I will start a new job in a couple of weeks. So this is the last shot, and I'd like to make it interesting, to make it good. So feel free to ask questions, to interrupt me at any time. And I'd like to describe a bit the workflow of fNIRS and how to structure data and to make them informative can be helpful and useful in a center like CIMeC is. So a multimodal center where many imaging techniques can be put together and are actually physically together and close to each other. And at the same time, I would like to share my experience and to share something that I did recently that may work and may be pointed towards this direction. Okay, so as Vittorio said, thank you very much, by the way. I worked most of my time in medical imaging from the physical point of view. I am a physicist by training. And today I would like to talk to you about fNIRS, a little bit of introduction, how it works, how it is, which are their physical principles, how multimodal imaging with NIRS in particular can be fused, how I think data should be structured in the fNIRS environment, how I think data should be visualized in order to get insight from them in the fNIRS environment, and how data can be fused between different modalities. I will try to share with you my idea of fusion, which is something that I developed in my personal experience. Okay, so let's start from the beginning. What is fNIRS? I believe most of you already know. Let's give it a quick recap. It's all there in the acronym. fNIRS means Functional Near-Infrared Spectroscopy. It's a spectroscopic technique. It means that it uses light. It makes use of light. It uses near-infrared light, so a very specific part of the electromagnetic spectrum. And it's a functional technique. It means that it's not measuring anything structural or anatomical, but it's measuring something related to the activity, to the functionality of the human body. In particular, concerning neuroimaging, fNIRS measures something which is related to the brain, especially. And it may also be used for measuring the activity of muscles or other active tissues, and it's been done in the past, especially in sports science. My main focus is neuroimaging, so I've used it most of the time for neuroscientific purposes. So the difference between anatomical imaging and functional imaging can be quickly pictured by these two images, three actually. Anatomical imaging has the purpose to visualize geometrically the structure of a part of the human body, or a body. In this case, the head is this one is an MRI image. Is very precise. Is very contrasted. Is very resolved, and it gives us the geometry. It's a static geometry of the human anatomy. Functional imaging maybe or maybe not imaging as at the very end. Actually, is the measuring of an activity, usually over time, sometimes over space. And I presented two examples. This one is a time series, is a collection of time series measuring some sort of activity over time. This one is measuring some sort of activity which is color-coded over space. Okay. Why fNIRS works? It works because the activity of the brain is related to the neuronal activity, and neurons are cells that consume energy. In order to stay alive, they need to build energy, and this is done through cellular respiration, right? Cellular respiration uses many metabolites, and among them, oxygen. Oxygen is transported by blood and ultimately withdrawn by blood by the neurons from the blood, okay? And what happens to blood when oxygen is withdrawn from it? Who knows? Blood changes color. Blood gets darker, right? Venous blood is darker than arterial blood. And this is the main reason why we can use light to contrast between oxygenated region of tissues and deoxygenated regions of tissues. We can resolve venous and arterial blood by eye. A trained eye can quickly distinguish the two types of blood. Of course, we can build much more sensitive instrument, and we can be much more sensitive to small differences in the amount of oxygenation. The way we make this work is by using light sources, usually lasers, so controlled light sources, through which we can shine light into the tissues, and then we can retrieve how much of this light is actually coming back from the tissue itself. The amount of light that is missing, so the percentage of light that is missing, is related to the oxygenation of the tissue in the neighborhood. Okay, so technically speaking, this is a spectroscopic technique, and in particular an absorption spectroscopy technique. So what we basically do is absorption spectroscopy within living tissue. The way this is done is by putting fiber optics that deliver light and collect light, and we put fiber optics onto the scalp. Millions of problems. We have hair. We have to make them steady, stand still. We have to build cups. We have to overcome technical issues, but this is the main principle. We have to deliver light into the tissue and to collect the light that is diffused back from the tissue. This is a little bit dark, but those are what actually is the same. Is one example of an fNIRS study. So solution to deliver light onto the head of a newborn. In this case, this is a 1-day-old female. Actually, a very good subject. She was very quiet, and she was sleeping actually during this experiment. She was so small that even the smallest cap that I had wasn't fitting her, so I had to keep the cap with my hands. And in this case, we were monitoring her sleep, so the neural activity during sleep. The way this study, and usually those studies are performed, is by taking advantage of having more channels that we can distribute over the scalp. So in order to measure the activity over many spatial locations and different places on the head. And of course, do this over time. One of the advantages of spectroscopy is that it's very little invasive, so we can keep it for as long as you want. I mean, as long as the subject is not complaining about it. Okay. But it doesn't hurt. That's the message. This is generally what you get from an fNIRS machine. So you have a huge collection of numbers which represent physical quantities, and what you need is not just numbers. You need more than this. You need semantics. you need topology, you need to know what these numbers are meaning and how they are collected, how they are distributed, and what they mean at the end. So for example, those tables that I get represent time in rows and many channels or multi-channels or space, call it whatever, over columns. This is structure. Okay? So this is a very simple but informative type of structure of the data, and this is one thing that I want to talk about today. This is the main thing that I want to talk about today. Structuring the data, so to keep data organized, to give them a broader meaning than just the numbers that you get, and to give them ultimately to be able to retrieve more information from them by keeping both the quantities, so the numbers themselves, but also the information that they may carry, so the structure we can build upon them. So this is the main topic of the of today's seminars. Just a quick example from my background, so physics. For a physicist, lection I think 101, the first day they keep telling us a number is nothing without the unit of measurement, right? This is the first thing. This is the first structure that you can give to a number. It's something more that you need than just a number than itself, and that carries a meaning. Okay. When structuring data, you need to put together many things. You need to think about what data means. So you need an intellectual design. You need to plan your activity. You need to have the big picture in some broader sense. You need to be able to implement this, so to translate this into something, into a tool, usually a software tool, because those data are many, so you cannot handle them by yourself. You need software that helps you. And you also need maintenance, because structure and information carried by data may evolve in time. So science is not carved in stone, so you need, this is everlasting. So in particular, within the field of functional neuroimaging and fNIRS as well, the three main actors in the play are functional data, call it the protagonist, okay? You need anatomical data, so where do you get this functional data? Locations of the optodes or whatever. And you also have metadata, which are usually related to the subject, to the conditions in which you perform the study or the experiment, whatever, the type of the machine, how many data you collected, the shape of the data, and so on and so forth. But there is more. There are not just three separated and independent actors. In structuring data, you need to build relationships between the data. So the way those three actors actually speak to themselves, and this is the intellectual design that you need to plan when you build a structure in which your data may fit. And this is one example. It's an example that I took from the literature. It's a proposal to structure, in this case, MRI data, functional MRI data, which are usually coming from an MRI machine, in form of DICOM files or piles of images that you get on your pen, USB pen, somehow. The proposal is an organization, very simple, in a folder tree, which is informative of the content, and which is replicable, shareable, and hopefully used in many locations in order to be consistent. In this particular case, which is called brain imaging data structure, you can find it online at this website. The suggestion is to create a folder tree, to use the nifty format, which is fairly diffused, and to put together both anatomical and functional data, and metadata as well. This one, and I jump into NIRS now, is my proposal and my idea. During those years here at CIMeC, I developed quite a bunch of lines of software, and I tried to collect them into a single container, which is thought as a structure upon the fNIRS data. I tried to establish all the actors that are there, so functional data, anatomical data, and metadata, and to establish the relationship between them, and at the same time to provide the user the experience that is usually there when processing functional imaging data, together with a way to plan studies, organize the studies, storing the data, process the data, visualize the data, and to keep functional and anatomical data referenced between each other. So for example, when performing an fNIRS experiment, usually at the beginning you have to plan the experiment. So you have to decide where to put your fiber optics onto the head, strictly speaking. This tool provides you a way to simulate the distribution of the optods onto the head, and at the same time to simulate where the light is going, so which brain areas you are sensitive to, okay, or which brain areas you are not sensitive to, okay. So this is usually called probe design in the fNIRS community. So the second step is to organize the data. So let's say that you have planned the study, you have designed your probe, and you have started collecting your data. So you have tons of numbers in those text files. What you want to do is to import them in your software and to categorize them. My idea of keeping a data set organized is very similar to the idea of bits that I showed you a couple of slides ago. So to organize them into studies. Studies contain measurements or measures, and measures contains analysis. It means that you have a raw data, which is an analysis, which is contained in a measure, which is contained in a study. And you can develop the analysis by applying processing methods and to further develop the information that you get. What you usually want to do is then to replicate these development processes, so to apply the same process to many raw data, to many analyses, and this software is able to perform all of this stuff and to keep your dataset organized in a non-destructive way. So there is always time to get back to the original data. At the same time, you can organize your anatomies, so your anatomical references that may be specific anatomies of specific subjects, but also anatomical atlases, for example, if you use them. And at the same time, you can organize your probes, so your optos distribution onto the head, in order to use them and to relate them to various studies. At the same time, within the software, the software builds behind a tree structure which is practically identical to the bits one. So you always have your files organized that you can share with other people, and you can retrieve in order, for example, because you want to use them with a different software. Okay. Then I developed methods. Methods are mathematical operation that you apply on the data and to get information from them. They can be pruning of data, so thresholding and say this is good, this is not good, and I want to get rid of them. It can be spectral filtering, it can be time filtering, they can be trial averaging, they can be more complicated, they can be three-dimensional optical reconstruction of the data. So let's say that you have an arrangement and you want to triangulate your measurement in order to get something which is a volume information, not just a point-like information. So we implemented methods and mathematical operation that you can apply to your data. And then at the end, we build viewers for the data. Two in particular. One is a two-dimensional viewer. It's a fairly classic time series viewer. We like to keep it cool, so it's interactive. You can click pretty much everywhere and get things, and also many colors. So I'm trying to advertise this software, but at the end I will give the bad news. So wait a bit. The viewer is pretty much like an EEG viewer. I'm sure you know it. So you have the time series, you can navigate the time series, you can zoom in and out, etc. And then there is the 3D viewer, so a space or a window, call it whatever, in which you can reconstruct three-dimensionally your data by making use of forward simulators of light propagation in the digital human head. This is a real data, for example, that I collected with a probe, which is depicted there with the red dots, and the blue and yellow points that go on and off are related to the activity of the brain. Okay. This one is making use of OpenGL. It's also interactive, it's also dynamic. You can monitor at the same time the space and the time. Okay, so before doing this, the bad news, I promised that. It's written in MATLAB. I'm sorry. I know. It wasn't my first choice at the beginning, but it had to be at the end because most of the community is using it. MATLAB is pretty much used by academia only, I think, but it's heavily used in academia. So it's just a tool, and I think that it's not the tool which is ultimately deciding what's best. It's the craftsmanship that you can, if you have this, you can use any tool. Okay, MATLAB is good enough, so we decided to use this. This tool is early in its development, but most of it is there, and it's already online. It's on GitHub, and this is the web address. So I don't have, and I will not have many time to further develop this. So if anyone wants to step in, raise your hand. Not now, maybe also in the future. But this GitHub address is the right place where to get in touch, to discuss, to download it, of course, if you want to use it. It's still missing the documentation. I know, I will do it. I will never do it, but I will try to do it. Okay? So feel free to get in touch for this. Okay. Why did I do and why did I spend so long in developing these tools? Because I believe in data fusion. And to me, data fusion is not just juxtaposition of information, for example, functional and anatomical, because I can say, this is my functional data, and this is my functional change over time of my physical quantity, brain activity, okay? And this is the place where I record it. So what I do, I look at both of them, and I try to fuse the information within my brain, which is the ultimate data fusion place. Okay, but I can do better. I can try to relate the information and to visualize the information in order for this data fusion to be more informative, to be smarter, and to be better engineered. Okay, so to me, data fusion is something in which 1 and 1 makes more than 2. So it's not just a mere juxtaposition of information. The tools that I prefer and that I try to use to achieve this are tools coming from data science and data visualization. So trying to put together numbers and experience. Okay. Of course, you also have to keep in mind that at any time that the real fusion happens within your brain. So you only need to prepare things, but the fusion of information is there. So you have to prepare things in order for the information to arrive maybe in a quicker way, in a better way, but fusion ultimately happens there. And why do I think this seminar is important? Because CIMeC is fusion. This is the place where fusion should take place. We are a multimodal center. We have many techniques, both anatomical and functional. And to me, this is the best place where these tools should be developed. We should talk more, maybe. I should have come more to Mattarello in the past. And so this is what I think. I mean, I'm not kidding. I think the multimodal center have a competitive advantage in this particular case. It should be our mission. Fusion is our mission. Should be our mission. Okay. I promised I would have been quick, and I tried to be. So I would like to have a discussion on this, if possible. No, I'm not getting sentimental, don't worry. Just thank you very much. I've never been sentimental. I would be faking. Any questions? I open the questions. This is not for you, it's for the people from all over the world that are following us. Who's first? You're first.
I have a couple of questions. So the first one, okay, you talk a lot about fusion, and you showed an example. What other examples do you see easily following from the one that you have shown here?
Well, of course, in my experience and the world that I take information from is the world of neuroimaging. So in my idea, fusion is ultimately anatomy, so structure and function. At the same time. Of course, my example was in fNIRS, but fMRI has fusion. Okay, it's a simpler one because they both information are coming from the same machine at almost the same time, so it's easier there. MEG needs fusion.
Yeah, even related to the specific case in which you worked, neonatal, perinatal. For example, diffusion MRI is part of the picture. Could be part of the picture.
Part of the picture. Fusion may be not limited to a couple of techniques, maybe extended to more than two. But in that case, the equation 1 and 1 makes more than 2 may get more complicated, but you get the idea.
Okay. No, yeah, I was wondering how complex could be, because it's from the software point of view, but also from the process point of view of having, let's say, multiple recording, maybe not at the same time, but eventually.
Absolutely. It's, you're right. It may get really complicated in a snap. But that is the data scientist's job. So to keep things organized and to provide structuring of data. So provide the relationship between data. It's all in the relationship, right?
Okay, and I have one last question, which is very technical. You say that, unfortunately this is in MATLAB, and so maybe you have already in mind something else. So what, since you mentioned about 3D graphics, DB, and so on, what would you suggest as if you have to restart, let's say, given the same audience, what would you do for your tool?
Well, I have been both a MATLAB and a Python user extensively, and I spent periods of time in which I thought MATLAB was better, and periods of time in which Python was better. So I think that now I'm achieving fusion myself. I think they're just tools, and whatever is better. I thought MATLAB was better, and I still think MATLAB is better because the community is able to use it.
Okay, even though, let's say, software for 3D visualization, integration of data, usually they are not even MATLAB, Python, they are all C++ or other kinds of technology. Right. Maybe not for good reasons, but in practice they are. Like FSL, TrackBiz, Matrix, MRU, and whatever.
At the same time, I thought that I didn't want to compile my software, and I want to keep it open. So I excluded C and C++ at the very beginning because I wanted the software to be open.
Can be open anyway.
Sure, but it can be more complicated. So I decided to. MATLAB is quicker in achieving fast results, and so my choice was already at the very beginning between MATLAB and Python, actually, and I chose MATLAB for historical reasons, probably. It's good, MATLAB is good.
Yeah, then when it's about, let's say, clinical application, for example, it can be a problem because maybe, I don't know, the clinic may not buy licenses, and then what you do? And that's the usual problem.
You're right.
Or other industrial and so on, they don't buy licenses.
There is the option to use it with the runtime, which is there, and or we can refactor the code. I don't want to refactor the code.
No one.
It's like 80,000 lines of code. Not much.
So it's small.
It's
sorry.
No,
but technically it's small. It's below 100,000, so it's small.
So it wouldn't be a horror.
Yeah.
Of whole refactor. Any questions?
Not sure about my question, but anyway, I'm wondering what's going to be next. So you presented in your presentation, you fused data from structural anatomical data and fNIRS data. I was wondering if it's possible to fuse, for example, data from fNIRS and EEG, but considering that the two methods are actually acquiring information from different systems, if it's possible to integrate it, so considering the relation between blood oxygenation and electrical activity of neurons, if it's possible to produce a third signal that's the integration of the relation between these two, something like that. So
absolutely yes. It's called neurovascular coupling, and it's a very specific thing, and it's been done in the past.
okay.
So I'm not sure how much it has been fused in the sense of my definition, but for sure I'm aware of acquisition, contemporary acquisition of EEG and fNIRS data, which have been presented at the same time, let's say. Not sure how much they've been fused in my definition. But yes, it's all in the relationship between the data. Of course, the relationship between structure, anatomy, and functional is one type of relation. A relationship between two functional data is a different type of relationship, but it may be there. We can grasp it at some point.
Do you think that, for example, in my project I have EEG and structural MRI and functional MRI. Do you think that this kind of fusion can be reached by something like me, so the scientist that does the EEG part, or you need a data scientist to do this kind of fusion? Because it sounds so it sounds beautiful, but sounds so complicated for me to find a way, a proper way to fuse functional and structural data and EEG data and maybe other kind of data in one signal. Let's say
well, strictly speaking, I believe it's the job it's
But
the job of a data scientist, and it's not the job of one single person. It's something that can be done together, and it's usually more something related to probably to physics or something like that than or yes, computer science than neuroscience. It's a hard job. Ben?
Since you're near all of these animal models, I was wondering if any possible fusion there had come to you, or if you proposed any to some of your colleagues that they stand, or just because they're using some different techniques also than what we met up here.
Well, my examples were, of course, taken from my personal experience, which is in humans. But I see no problems in moving to a broader species container. So should be the same. Maybe dimensions may change. For example, you may have physical constraints. Light diffuses with a certain pattern. So doing NIRS in animals may be not that easy. It's been done. It's been done in dogs for sure. It's been done in mice and smaller animals, but using open skull, so no diffusion. So you may have technical differences, but I don't see conceptual differences. Should be something.
Does the software incorporate databasing capacity, and do you see databasing as an essential tool for sharing big data across labs and centers?
It does at a very basic level, so you can import, export, move things around, and for example, move measurements between studies. I wouldn't call it a strong database software. Usually, what a basic user should need, it's there. And I used macro functions for it. So at a very basic level.
We'll have a quick question. I think I really appreciate the position of, let's say, database spot within the schema of your software, which is pretty central, I would say. My point was whether the fact that you know that you're going to arrange the data in some way, and that for the software, for your software to be like functioning at its best, you need several information. I was wondering whether this shapes up your experimental routines as well. So I think that I need information, so I modify my research routine. For example, I before this software, before this data structuring, I was never interested in, I don't know, the number of whatever diodes within the cap. But then I include information and I modify. So I'm doing different kind of science according to different data structure. That would be my point.
So a real feedback of my software into my lab experience has never take place because I just put it together in the last month. So what I can give you is my previous experience of that. Yes, the way I handle the data has always informed on the way I collected them. So it's something that grows together. I'm not sure if I answered your question, but
yeah, pretty much. I mean,
yeah, it does.
Yes.
And also my background, of course. It's there together.
I mean, one of my points is that if you think that your data are going to be processed by, let's say, a human being, then several things you just don't care about. But if your data they are going to be processed first by a machine, then several details, then you're forced to include them into your, for example, experimental routine, or just, for example, to put some layers between your cap and your computer, because you need, for example, to record version of the caps and so on. So that would be my point, like modifying your research routine to have richer and more structured.
That's already a feedback, for example, and I'm sure that with more experience, every one of us would have more. I go back to the same point. The way you collect and the way you process the data, if this is done by the same person, as you were saying, but also if it's done within a group, can be informed. And this is another advantage of a multimodal center. So we can talk to each other, and you can say, I need this tool because I would like to fuse this data. You may get the idea, but you may miss the way the idea can be carried over. So it's probably something bigger than a single human being, but it's something that community can achieve, both in one sense and in the feedback. Domande da Milano? Però è scritto lui.
They actually, I don't know if they're actually asking whether you can either repeat or rewrite the link to your GitHub page.
Sure. Well, also the presentation can be shared, so it's everything there. Also, I understand pictures are not large. You can enlarge them with your computer and enhance the experience.
Okay.
Can call it a day.
No more questions? Okay.
Thank you very much.